All right, this is lecture 4, part 2. We're going to finish up our lecture on DNA by talking about protein synthesis. So you should have already watched part 1, which is mitosis and meiosis, or cell division, and the structure of DNA, and also how DNA copies itself, or the process of DNA replication. So just as a review of DNA structure, DNA is made up of a phosphate backbone with nucleotide bases that are labeled by these letters T, G, C, and A. And there are two strands to DNA. The coding strand, which is a 5' to a 3' direction, and the non-coding strand, which is complementary based on the base pairing rule where A's and T's bond together and G's and C's bond together, and the complementary strand is a non-coding strand that runs in the 3' or hydroxyl end to 5' phosphate end direction. So all of these nucleotide letters, the A, G, T, and C, make up the code of DNA. And this is the way that the code works. A sequence of 3 nucleotides codes for 1 amino acid. And remember that amino acids are the building blocks of proteins. So we can say that 3 nucleotide letters are one single amino acid word, which will be translated into an amino acid. DNA then is one long strand of nucleotides that codes for strings of amino acids. That is, the coding strand of DNA is a string of nucleotides that codes for a string of amino acids. One string of amino acids grouped together in a single gene makes a protein. So we put the words, which are the 3 nucleotide letters, together to make a final protein, which is like putting sentences together in a word. Chains of DNA nucleotides then are protein information codes. So the purpose of DNA is to hold the instructions for synthesizing or making proteins, and those instructions are written in nucleotides. So a sequence of 3 nucleotide letters codes for 1 amino acid word, and this is called a codon. A string of amino acid molecules then makes a macromolecule protein. There are 64 different codons in the genetic code, and those 64 different codons code for 23 types of amino acids. So in other words, there's a redundancy to the code. There are multiple codons that code for a single amino acid, and there's 23 types of amino acids. Now, if this was a biochemistry class, I would have you guys go through and memorize all the different codons that encode for each of the different amino acids and the basic structures of the amino acids. In this class, we're not going to do that. In this class, I just want you guys to understand the way that the genetic code is written. In addition to that, there are start and stop codons that indicate the beginning of where a gene should be read, and the end, or the stop codon, is where a gene ends. It's like the capital letter at the beginning of a sentence and the period at the end of a sentence, and that's how you know what to read. So here's the genetic code. So you will notice over here, these are the amino acids: phenylalanine, leucine, isoleucine, methionine, which is also the start codon, valine, serine, proline. These are all amino acids, which you guys do not need to memorize. And then here are the 3 nucleotide bases that encode for that amino acid. And notice there are multiple sequences that encode for a single amino acid. Now that's handy if we have some genetic point mutations, which is what we talked about in the previous part 1. So U, nucleotides code for phenylalanine. UUC also encodes phenylalanine. Now, do you notice anything interesting about this genetic code? What are the nucleotides for DNA? A, G, T, and C. Do you see any T's? Nope. We only have U's. So which nucleic acid is made up of A, G, U, and C nucleotides? RNA. So the genetic code, where we're actually reading amino acids, is written in RNA codons. So what we need to do to get from DNA to protein is to use RNA as a translator. So to make amino acids strung together as a protein, we need to use the RNA codons. To get the RNA codons, we have to read the DNA. So did you notice the codons contain the amino acid uracil and not thymine? That means that RNA is the basic genetic code. So the actual expression of the genetic code is to read the DNA first, convert it into RNA codons, 3 nucleotide bases at a time of RNA, and then read the RNA to make protein. So it's as if the DNA and the RNA are 2 different languages, and they need to be translated in order to make the final language, which is a protein. So protein synthesis is the process of making proteins by reading the DNA genetic code. DNA is an information storage code, and the actual creation of a protein from the DNA allows for the expression of the traits. So we've talked about traits such as eye color, pigments, other things that are the actual expression of us as individuals. So this requires DNA, RNA, amino acids because they are the building blocks, also ribosomes, and several enzymes in order to make a final protein. There's 2 main stages of gene expression. Get out some notes. We're going to go through each one, talk about them, and draw them out. Now, one thing I want to say here is that very often students mix up transcription and translation with what we previously went through, DNA replication. So make sure that you get a good handle on DNA replication first and the enzymes and steps involved for DNA replication. That happens before mitosis so that cells can be copied. That's very different than transcription and translation, which is happening all day, every day in our cells in order for cells to function to make proteins that they need for their everyday functions. So an overview of gene expression in a cell, in an animal cell, such as a human cell. We're going to start with DNA in the nucleus. There's the coding strand and the non-coding strands of DNA. From the DNA, we're going to make mRNA. The mRNA will then leave the nucleus. And from the mRNA, we will make a final protein. So overall, we're going from DNA to mRNA in the nucleus. And then outside of the nucleus, we're going to go from mRNA to the final protein. So we're first going to talk about the process of transcription. Transcription is the process of making RNA from a DNA template. So the RNA that is being made is called the messenger RNA or mRNA. mRNA is linear, just a single linear strand of nucleotides, and that requires an enzyme called RNA polymerase. The mRNA can then leave the nucleus for the next step. It's very important to know that one of the major reasons that we use mRNA rather than DNA to make a protein is because the DNA needs to stay in the nucleus unchanged in the process. Why? Because we don't want any mutations or changes in the DNA that could lead to some major changes in cell function and even death of the cell. So the DNA has to stay unchanged in the nucleus, and the mRNA will be the middleman that will transcribe or make a script of the DNA to tell the cell what to do to make a protein. So this is the basic process of transcription. I'm going to show you this diagram and then we're going to draw it out. Transcription is taking the double helix of DNA, unwinding it, opening it up, and adding RNA nucleotides in a complementary fashion to the template strand. Okay. So the template strand is the 5' to 3' strand, and we're going to add mRNA nucleotides based on the complementary base pair rule, except that there are no T's in RNA, so where you would normally add a T, namely with an A, we would instead add a U. So here we have the mRNA being made, and from the template there was an A, so the mRNA added a U. Here the template had a C, so the mRNA added a G, etc. G to C, A to U. When you see a T on the template, then we add an A. All the way through until the entire gene or the sequence of nucleotides for a single protein has been transcribed. Now we call this transcription. This is making a script or going in, reading the DNA, writing it out in the mRNA code. When that process is done, we will close up that segment of DNA and rewind it back together. Okay. So let's take a second to draw out transcription. Okay, transcription happens in the nucleus, and it is taking the DNA template and making an mRNA transcript. So we have the 5' to 3' coding or template strand of DNA, which is wound up with the 3' to 5' strand of DNA. We need to open up a segment that we're going to copy, and we will use an enzyme which is RNA polymerase to copy that segment of DNA. We're going to go on the 5' to 3' segment, and we will copy one by one, nucleotide by nucleotide, creating an mRNA linear strand. So we're going from the DNA template to a segment that has been copied into mRNA. So the mRNA will contain bases A, G, U, and C, and it's going to be linear. It requires RNA polymerase again, and once it's done, the mRNA can leave the nucleus. That transcript then can be used to make a protein. So that is transcription, where we're taking the template strand and making an mRNA message. Now we're going to take the mRNA and turn it into a protein. So notice transcription translation, very similar words. So the way that I always think of this is that you have to take a script and read your script before you translate. So we make a script in the nucleus, which is the mRNA, and then we read the script to make a protein, and that's translation. So the script is in nucleotides, and the final product, the translation of that product, is the protein. So there are several steps to translation, and we're going to go through each one of them. So during these steps, in addition to the mRNA that we just made through the process of transcription, there are two other types of RNA that will be used during translation. So the mRNA holds the genetic message from the DNA that was made in the nucleus. We will also have ribosomal RNA and transfer RNA. So the mRNA leaves the nucleus and attaches to the ribosome. Remember, back to our organelles and our cell lecture in lecture 3, we talked about ribosomes as the site of protein synthesis. Ribosomes are actually made up of RNA, large subunits and small subunits of RNA. So ribosomal RNA has two functional pieces, the small subunit and the large subunit. Both have to be bound to begin the mRNA translation. So what happens is the mRNA leaves the nucleus and binds to a small subunit of rRNA, which will either be, where are the ribosomes? The ribosomes are either free-floating in the cytoplasm or they're attached to the rough endoplasmic reticulum. So the mRNA is going to come in, bind to the ribosomal RNA, and then once it binds to the ribosomal RNA, then the large subunit of ribosomal RNA will come in. We'll have a functional full ribosome that we can now use as the stage to translate the mRNA transcript. So mRNA is going to move through the ribosome in a stepwise process through 3 sites on the small ribosome. There's an A site, a P site, and an E site. And it looks like this. So the mRNA attaches first to the small subunit. Once it does that, then the large subunit will come in and we'll have a fully functional ribosome. On these subunits, we have an A site, a P site, and an E site. And again, you want to read this kind of like our previous DNA replication diagrams. We're going to read this from right to left. So the mRNA is going to come over here at the A and P site and then move in this direction towards the E site where it then will exit. The tRNA is going to actually transfer and bring in amino acids. So the tRNA is a long strand of nucleotides. Again, it's RNA, so it's made up of nucleotides, but it looks like a little hairpin. We don't use hairpins a lot in our generation, but look through your grandma's dresser and we have hairpins that kind of have this little T shape. So it's a hairpin-like folded structure. Each tRNA contains complement bases to a codon called an anticodon. So remember that mRNA has the 3 bases, A, G, U, and C combinations that make up the genetic code. Each triplet code, what did we say? U, U, U encodes for phenylalanine. So we'll use that as an example. So if the mRNA is U, 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 then the tRNA is going to have the complement bases to that. So the anticodon on the tRNA, what does U bind to? U binds to A. So if the mRNA is U, 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 the tRNA is going to have A as its anticodon. So it knows then and has a piece of the tRNA that will dock onto the mRNA. It'll attach to the mRNA and bring in amino acids. There will be enzymes in the cytoplasm of the cell that will help to match the correct amino acid to the correct codon or anticodon sequence in the tRNA. So this is that hairpin, and we don't have hairpins that look like this anymore, but this is old school, guys. So here's the hairpin structure of tRNA. At the base of it, we've got our anticodons. In our example, let's say it was for phenylalanine. It would be AAA, which is the anticodon to UUU in the mRNA, which encodes for phenylalanine. Then there are enzymes that will attach phenylalanine to the tRNA so it can bring the tRNA into the ribosome mRNA complex. So here's what we're going to do. I'm going to read this and then we're going to draw it out. This is quite complicated, so bear with me. Take some time to draw this out for yourselves too, guys. So first, we're going to start with the start codon. The start codon is going to start at the P site to get the whole process beginning. The next thing that happens is that a tRNA will come in and attach to the A site at the ribosome. At the A site is where we get the first attachment of the tRNA with its amino acid. The tRNA will then move one side over. As the mRNA moves one side over, they will march together, move over like an assembly line to the P site. At the P site, amino acids will be bonded together. So this first amino acid will wait, and a second tRNA will come in, attached to the A site. That A site amino acid can then bond to the amino acid sitting in the P site. That peptide bond will form between the first and second amino acid, and then everybody will march one site over. That first amino acid will then move to the first tRNA will then move to the E site. The second tRNA will move to the P site. And a third tRNA will come in and add a new amino acid to the A site. That third tRNA that's in the A site can now peptide bond to the second amino acid that's in the P site. The first amino acid has now made its peptide bond with the second and will exit at the E site. I know. Let's draw it out. Okay, so we have this is translation. We have the mRNA transcript from the previous step, which was transcription. It left the nucleus and is now out either in the cytoplasm or going through the endoplasmic reticulum. It finds a small subunit of a ribosome and it attaches the start codon at the P site. So the first 3 nucleotides are the start codon. They will attach at the P site. The ribosome large subunit will now be fully assembled and translation can take place. So the first thing that happens is that we have a tRNA that recognizes the start codon and will add the start amino acid. Then our first actual coding tRNA will recognize the first amino acid sequence, or the first codon, and what do we say? U. So there's U at the A site, and the tRNA is going to recognize that because it has anticodons A, and it knows that when it sees a U, it's going to bring in a phenylalanine amino acid. So we start by attaching our first tRNA at the A site. So A is for attach. I don't know if they're actually named that way, I just call it that. A is for attach. We have our first attachment here, and then everybody's going to move one over. And now I'm not going to draw the ribosome anymore. I'm just going to draw the A, P, and E sites. So now everybody moves one over. A, P, E site. Now we have our mRNA start codon is now at the E site. Our first coding, phenylalanine, is at the P site. And our next coding has entered at the A site. The first start codon added a start amino acid with its tRNA. The next added phenylalanine based on its tRNA, which was A. And those peptide bonded together at the P site. Now we have a third codon. Let's say it's G. I don't know. Make it up. GGG. GGG will be recognized by another tRNA. And what bonds with G? C. So that tRNA will be C for its anticodon. And it's going to bring in amino acid number 3. So amino acid number 3 is now in the A site. Everybody can march over one more. When everybody marches over one more, then we have amino acid number 3. Our first phenylalanine, which is now bonded to the methionine, or the start codon. And number 3 is now at the P site. That leaves room for a new amino acid to come in based on the mRNA code that is continuing to march through this assembly line. When amino acid number 3 moves to the P site, it will bond a peptide bond to the phenylalanine. Now we have a fourth amino acid that would be brought in based on the codon and the tRNA anticodon to the A site. Again, it will continue. That will march over. Peptide bond at the P site. Now that the peptide bond is between 3 and the phenylalanine, that will also march over one more, and it will exit at the E site. So the A site, we attach. The P site, peptide bond. E site, exit. And the mRNA will continue to move through until one by one the codons are matched with amino acids. The amino acids are peptide bonded, and then the tRNAs exit. Okay, so attach, peptide bond, exit. The tRNA will bring in an amino acid to attach. The tRNA will bind two amino acids together, make a peptide bond at the P site, and after the peptide bond is formed, the tRNA will be released and exit at the E site. We'll keep repeating this until we hit the stop codon at the very end of the mRNA transcript. When that's done, the ribosome and the mRNA will break apart and the protein will be released. Now, if you don't like my drawing, let's go through the drawings that we have here. So here's our start codon on our mRNA. Here's the mRNA docked onto the fully assembled ribosome. The start codon is attached at the P site and waits for the tRNA to move in to the A site. The first tRNA then, after the start codon, attaches at the A site based on the codon anticodon match and brings in an amino acid. And everybody marches over one. The first amino acid can bind to the start codon amino acid, and that tRNA can leave, and then it will wait for a new tRNA to come in and attach. The new tRNA comes in at the A site. The A site tRNA is now in place to bond to and form a peptide bond with the P site tRNA. The P site tRNA will peptide bond, and then everybody marches over one and exits. So the ribosome is going to guide the translation process and hold the mRNA and the tRNAs in place as each amino acid, one by one, is bonded to make a polypeptide chain. The polypeptide chain grows and grows until we hit a stop codon and we have our final protein. Okay, did you guys get it? If not, the nice thing is you've got a video, right? You can go back and review. So remember, attach at the A site, peptide bond at the P site, exit at the E site. So think of it like an assembly line process. One by one, codon by codon, the mRNA will march through and attach the appropriate amino acids together with the help of the tRNA. So as an overview, we already talked about in part 1 the process of DNA replication. We've now completed the process of reading the DNA to make a protein. First through transcription, which was making the mRNA from the DNA in the nucleus. The mRNA can then leave the nucleus to make a final protein in the process of translation. There is a video that I recommend, and I will link to our site, which also shows transcription and translation. So overall, I want to make sure that you guys don't mix up, because I see this happen all the time on the exam, that you don't mix up replication, transcription, and translation. So replication is the process of making sister chromatids. So we take one strand of DNA, copy it, take another strand of DNA, copy it. We end up with two identical sister chromatids, or copies, of DNA. Then the cell can divide and reproduce, and that happens during both growth, development, and in everyday cells where cells need to be replaced. Transcription and translation are happening all day, every day, in every cell as a basic part of making the macromolecules, proteins, that are needed for everyday cell functions. So transcription is the process of taking regular DNA in a nucleus, opening it up, and reading a single gene to make mRNA. The mRNA will then leave the nucleus as a transcript to be read. That transcript will be translated by the tRNA. And the tRNA is going to read step by step, marching through the ribosome, and add the correct amino acids one at a time until we get the final product, which is a polypeptide, many amino acids bonded together with peptide bonds to make a final protein. Those proteins will then be functional proteins in cells, and we learned about what the different proteins are in cells. So they could be channels, they could be receptor molecules, they can be any number of secretion products that a cell is making, many things that a protein can do. So one last bit is that I said, kind of simply, that the mRNA transcript is a single gene. Well, as it turns out, there are pieces of DNA that are coding and non-coding parts of a particular gene. So this is different than the coding and non-coding templates of the strands of DNA. This is actually in a single gene. There's something called exons and introns. The exons are the coding portions that will actually give a functional protein. The introns are non-coding pieces that need to be spliced out to get a functional mRNA transcript. So this is an extra level of control that we see in more complex organisms. So more complex organisms like us, eukaryotes, other animals and other eukaryotic cells, have exons and introns. These are sequence of nucleotides that are encoding a protein in the case of exons, and fragments of sequences that need to be spliced out to get a final mRNA. So in other words, the mRNA, actually there's a step between transcription and translation that has to happen to remove the introns. So they have to remove the introns to get a mature coding mRNA. This has happened through splicing of mRNA to form a functional transcript. Other things that happen to the mRNA to make it stable is that there's a 5' cap that is added and a poly A tail that is added to the mRNA. All of this just adds to the step between transcription and translation to both stabilize the mRNA, and you can make tiny changes to the mRNA to get minor changes in the gene before it's translated. So this is basically what it looks like. You have exons and introns in the DNA. The DNA is then transcribed into the mRNA. The mRNA will then be processed through splicing, where the introns are removed, and the coding regions will be spliced together to make the final mature mRNA transcript. So again, there are two main stages of gene expression, but there is processing in between the two. So we have transcription, which is DNA to mRNA, and that happens in the nucleus. Once that mRNA is created, there is processing that occurs to make a final mature mRNA transcript. That mRNA transcript is then ready to dock onto a ribosome and be translated into a protein. So DNA transcription occurs to mRNA in the nucleus. The mRNA is then processed, and the mRNA will leave the nucleus and bind to a ribosome. The mRNA will then be translated by the tRNA at the ribosome, either in the cytoplasm, free, or on endoplasmic reticulum. At the end, a complete protein will be released and the ribosome will disassemble. So DNA is transcribed in the nucleus. The mRNA will be processed and then leave the nucleus, find a ribosome, attach to the ribosome will fully assemble, and then the tRNA will come in and one by one add amino acids to get a string, amino acid chain or a polypeptide to form a final protein. There is some regulation that can happen to control protein synthesis, or also called gene expression. So the expression of the gene is to make a protein. So we can regulate the synthesis of proteins or the expression of genes in many ways. There are transcription factors that bind to DNA that can turn a gene on or off. There are activators that bind to DNA that can increase or decrease the rate or the speed of transcription of a gene. There are histones, which are proteins wrapped around the DNA. Remember, we talked about those with chromosomes, that can make it less accessible for transcription. There's also something new called methylation, and we're seeing a lot of this in research currently. DNA methylation is adding a methyl or a CH3 group to DNA. That can turn off transcription of a gene. Interesting too. Some indication in the current research that methylation may be inherited. RNA interference is another new area of research, shortened as RNAi. So RNAi is using antisense strands to mRNA to bind to mRNA and block it. It keeps the tRNA from coming in. It also causes the cell to degrade it, and that can prevent translation. So they're using RNAi actually to, in research, to prevent mRNA from being translated. The last is what we just talked about, which is mRNA splicing, and that is processing of mRNA can actually activate or inactivate the final protein that will ultimately be produced. So a lot of interesting pieces here in regulation of protein synthesis that can change after the DNA code. There are additional levels in addition to the basic code to change how genes are expressed in an individual. Okay, I think that's enough for today. That was the end of part 2 of DNA. Please let me know if you guys have any questions on either part 1 or part 2 of DNA.